Dan, take it away.
Okay, so since this is a diverse group, I'm going to kind of go through one area of research that I'm very interested in, which is the microbiome in general, which is basically a new area of research in which we now are really starting to associate the trillions of microbes that are now inhabiting us and everything around us as a way to not only understand disease and health, but also to understand how we transmit disease and also how can we then intervene and manipulate it. And then on top of it, other ways to be able to use it as a way to detect and be able to monitor health and disease. So the outline is basically, I'm going to go over what we kind of know about the host microbiota, which is basically the microbiome, which encompasses all the microbes. Then the idea of healthy. I'll go into some of the novel microbiome-based therapies that are coming out in ways to be able to regulate and manipulate the microbial populations to improve health and to also for diagnostics. And then some of the ways in which the environment then plays a role, which I think is the greater picture, which is now you have the greater environment that plays a role in the microbes that then play a role on your health and disease. So we really need to bring this out, and this is what David was talking about, using some of these new technologies to be able to characterize everything around us. So I'll go through kind of, since I get everyone on the same page, of kind of how this all came about. The first pathogens were really kind of described and discovered in the 1600s, and then by the early 1900s, we started to be able to culture them, and that was a big jump, because now we can actually culture the bacteria that we couldn't see before, and then we could start to characterize it. Then the next kind of revolution as far as I think is moving things forward was our ability to then sequence those bacterium and microbes, because then we could actually use the genes that it produces and understand more about the functionality of these microbes rather than just growing ones that, and also it gave us the ability to then look for and characterize microbes that we couldn't culture, which ends up being 60-70% of the microbes in your microbiota. But to me, really, what sparked it all, because the genomics was great, but you can only sequence one organism at a time, and you had to be able to isolate, and you had to know something about that organism to even get the sequence, was the invention of this next-generation sequencing, or second-generation sequencing, in which you are able to then sequence anything in the sample. You don't need to have a priori knowledge anymore. You can just take the sample, fragment it, put adapters on, and sequence everything. So every microbe, all of the human, everything that's in the sample gets sequenced, which to me was a real eye-opener as being infectious diseases, because I've spent all my time designing primers and things to design for specific pathogens and detect those pathogens. But if we didn't know what those were, so say I was working in Trinidad and we have, people get sick all the time and it's not dengue, well, what is it? It has to be something else, but we didn't know what it was, so we could never go look for it. This gives us the tool to be able to look for anything. So now I've taken this tool and sequenced lots of human samples to be able to try to identify etiologies of unknown origin where they don't know what is causing the disease. So that's really kind of my mainstay is I'm a virologist, and this was a tool to really expand the idea of understanding virology, but also understanding pathogenesis and disease transmission. So it was a real win-win. And it wasn't until I really got into this to start to understand all the diversity of microbes that are inhabiting us and how they could be impacting our health. So this kind of sums it up. Is we have about 22,500 genes. But our microbiome has over 1 million. We can be 99.5% similar at the genetic level, but our microbiomes can be up to 90% different. What that's telling me is that 10 people in this room can get infected with influenza, and maybe it's not your genetics that's driving it, but your microbiome. And it's those drastic differences that kind of give you an idea of maybe those are some of the subtle differences that we see in individual susceptibility to particular diseases. You have 5 to 10 times more bacteria than you do human cells. what I thought was really interesting is diving into this is that 10 to 20% of our human genome is actually endogenous retroviral sequence. So it's not even human. 20% of you is actually virus, and these viruses are actually producing transcripts and genes, and they are actually regulating our genome. So here we have now a trace of ancestral, a trace of viruses throughout time that can be traced through our genetics that then influences our genome. This is all microbe and has nothing to do with the human. And then more importantly, which I think is really the case, is the enormous diversity of bacteriophages that are not only in us, which is 100 times more than the bacteria, which makes it the most dominant organism that inhabit us, but also the most dominant organism on the planet. So yeah, it's really trying to encompass all of these. So kind of our players really are, we have the commensal bacteria, which I consider more mutualistic bacteria, things that are kind of co-evolved with us and keep us healthy. Then obviously you have pathogenic bacteria that cause disease. Then we have these bacterial phages, and we have endogenous retroviruses. So most of us are infected with CMV and all these other viruses that are latent viruses, herpes, and we also have tons of phages, because if you have bacteria, you have phages. Then there's the eukaryotic parasites, which a recent study just came out in mice that showed that a group of mice housed in different facilities had different immune cell reactions. So one had more immune cells than the other, and they didn't really know why. And when they infected them with the bacterium, one of them was susceptible and one of them was completely resistant. And what they found is actually one of the colonies of mice actually had a parasite. And the parasite was really the driver difference between the disease susceptibilities. So this is all like, until you can look for it, you don't know. And this really was what opened my eyes to when I started doing, because I did a lot of animal research, and yeah, these animals could be infected with other things that are really driving the disease profile that we see and not the genetics. So, and then of course we have fungi and then yeast, which I haven't put on there. But the reality is that we need to understand that these microbiomes that we've inherited through our moms and are actually genetically transferred from our ancestors, and these things are co-evolving. They can change due to our environment, and I think that that's really the grand player of this whole thing, is that it's not stagnant like our genome. This thing is fluid, so you can manipulate it in ways, and you can change it by changing your environment or changing your diet. And these are really what we need to understand, and I think that these could be major players in understanding our diseases. So that's kind of how I see people now, is not necessarily as a person, but more as a bunch of microbes. There's been a lot of press on this. The Human Microbiome Project was one of the big studies that came out of the NIH. It was huge. It was 200 scientists and 80 institutions. What they did was they sequenced a bunch of different sites of the human body, and they were able to then correlate those sites with different microbial communities. And the take-home kind of message really was that not only were these distinct communities, but they were distinct based on different body sites. So, and we've actually even done further research where we can even break it down even within the organ. So we've done influenza studies in mice where we've looked at different lobes of the lung, and there's actually different microbiome communities within the different lobes. So now when you're talking that kind of diversity, then you can start to understand how they could be playing a role in disease depending on what infection you get. So one of the things that they found is that there was significant variability between the bacterial species, between subjects, but the reality is that the species didn't really matter, because it was really about the function of the community. So what they found was that you could have very different species than me on your skin, but those species carry the same functions. So overall, the community serves a similar function. So that's what kind of breaks down the idea of classifying a bacteria. Might not mean that much if you don't know what the bacteria is doing. So that goes back to all this research right now is being done to classify bacteria, but yet we don't understand what those bacteria are doing. So now we're all sitting here in these talks, giving these nice little profiles. I could show you, hours worth of profiles of these microbiome profiles, but at the end of the day, we need association, right? And that's kind of where we're trying to drive to, and where some of these therapies could actually play a role, is to actually learning about the function of them and then learning how to manipulate the functions for our own benefit. Okay, so here's your definitions of healthy. we all think of healthy as someone who, has a good diet, who's fit, and who exercises well and everything else. But to this point, we really haven't really pinpointed, what determines overall health. And just because you feel healthy, does that mean you are actually healthy? And I think that this is some of the research that I've worked on that has really helped me believe that health is not necessarily, it's a state. It's not necessarily, and it's fluid. It's not necessarily, I am the definition of healthy. I can be healthy 5 minutes ago and I might not be, because I ate McDonald's, a Big Mac, I might not be healthy in a half hour. So it's really kind of understanding that dynamic as more of a state rather than, so there's lots of factors that influence this process. One of the big key ones is long-term stress, but they've been, environmental factors, your diet, your lifestyle, even your social interactions through the people you hang out with, and where you live, whether it's urban or rural. These are all things that have been shown to influence disease and health. But as far as the microbiome is concerned, it has been, over the past 3 or 4 years, there's been a ton of work has been done. And these are basically some of the diseases that have been associated. So we're talking everything from metabolic disorders to genetic disorders to psychological disorders. And not just about obesity anymore, cancer as well. So really it kind of brings this idea of, what I'm starting to incorporate in disease, which is really you have your host genetics, and then you have the host microbiome at that point, and then you have your invading pathogens. So whatever your disease state is when that pathogen comes in is really what's going to determine your clinical outcome. So if you have an inflammatory microbiome that allows you to be more susceptible to, say, influenza, or you have pathogens already in your upper respiratory tract, like Streptococcus pneumoniae, and you get infected with influenza, you're probably more likely to develop severe disease than if you did not have it. So it's really incorporating this extra layer. And I think this extra layer not only will help us define the differences between the individuals at the population level, but it'll also help us better define how to predict the outcome of clinical disease and what role these guys are really playing in disease susceptibility. Does that make sense to everybody? Is everybody kind of on the same page? Okay. If you guys have any questions, please let me know. Okay, so during my research of all this, what I really started realizing is that so we've always had this idea of the good bacteria and the bad bacteria, and we don't want the bad guys, right? Well, we kind of know who the good guys are, things like lactobacillus and even E. coli, but that's not A70157. But these things are ubiquitous. They're commensals. They hang out in our body. They're all over the place. And they tend to work with our bodies and our genes and our immune systems to kind of form a more mutualistic or symbiotic relationship. And then we know the bad players, because these are the ones that have always come up through time, and they wreak havoc. And we kind of been focusing all of our time on understanding the bad players. Not so much the good players, but always the bad players. And then another layer kind of came in with all this research, which is what is considered a bad player. Like, I know anthrax is a bad player, all right? It's got toxins, it gets in you, it causes disease. But then there's all these bacteria that basically, they live in us, they're everywhere, but yet in certain environmental conditions, they can be bad players. These are things like Helicobacter. These are things like, Pseudomonas aeruginosus. These are things that exist, but in the right conditions, they can actually become more pathogenic, cause disease. And I think that this is really the underlying part of health, right? Is that I think that a lot of people have these more pro-inflammatory microbiomes that are causing you to be more susceptible to other diseases because you're already in an infected disease state to begin with. And I'll show you some studies which we've actually done in monkeys, looking at the baseline of the monkeys before an infection and showing you they're already inflammatory. Like before they even, at their natural state, they're already heightened. Their immune response is already going. So this kind of goes into the idea of what a pathobiont is, and that we need to now recognize this new class of pathogens that are really, they're commensals, they live in a lot of us, but every once in a while they wreak havoc. And in certain ways they can dominate and wreak havoc as well over time, causing cancers, inflammatory bowel disease, these chronic diseases that we've been able to associate. And I'll show you some of the methods in which they've been able to counteract those things. But at the end of the day, how do we understand these guys? How do we classify them? And then how can we manipulate them? Because I think that this is an underlying layer that is not being looked at in the human clinic. In fact, I know it isn't. So this is actually a study in which we were actually looking at monkeys, and we were infecting with HIV. And really what we wanted to know is it was a microbiome study where we were trying to figure out how does the microbiome play a role in disease susceptibility to HIV. And what I found was interesting in the study is that we had one monkey that was very different than the rest of them. And so before I even did the microbiome on them, we didn't have any of these results. So this is kind of the baseline immune response, and these are inflammatory genes. And up here is the composition of the microbiome, okay? And what we were able to kind of show is that this guy reacted very different than the rest of these three, and to getting the disease. So how quickly it succumbed to the disease and how quickly it progressed to AIDS. So what we were trying to figure out is, well, at the point of infection, well, what happened? And what we found was that these three all had the same microbiome composition. It was all dominated by one type of bacteria, really, and it was Helicobacter. Okay, Helicobacter is actually, in a lot of cases, can be an inflammatory bacterium. So when we found, and then I did these guys, and this guy was very different. Well, what we found was when I basically did the baselines, we found that they're very different. One of these three guys are very highly inflamed. These are all upregulated inflammatory genes, and the blue is downregulated. So they're very different. They're almost opposite responses. And what it came down to was that this monkey right here got brought in from a different facility 2 weeks before the experiment. Was thrown in at the last second, and here you go. This is a completely different monkey, has a different diet from a different facility, and now you have a completely different response. Which then brings in the whole case of you're doing, I was looking at monkeys from all these different facilities across the United States, Yerkes, all of them, and guess what? They all had different microbiomes. So now the question is all these studies, do they really correlate if you have completely different systems, if this is playing a big role, which I do believe it is playing a significant role. So one of the fascinating things that got me involved really is the fact that the microbiome can influence vaccine efficacy and also viral pathogenesis, right? I'm a virologist at heart, so I'm not really a bacteriologist, but understanding the microbiome helps me understand the viruses. So I had to bring them in. I got my PhD in pathology, so I already understood the whole side of the host. So now I'm trying to bring in the host, the microbiome, and the pathogen to see if we can now make sense of it, which will probably just make it more confusing and more convoluted, but hopefully it will actually add to our understanding of clinical disease and be able to intervene, which I think we're making some strides. So these are a lot of the reports, everything from the vaccine. So they found a study came out in mice where they took a nasal influenza vaccine, and they basically administered it, and they found that it was flagellated, so bacterium in your gut that had flagella stimulated early B-cell progenitors to actually give you a more robust immune response. So what you just took in your nose, the gut is actually then stimulating everything downstream. So that's kind of really how much this is interconnected, and how much I think this whole gut-brain axis is really more important than we play it out to be. This is kind of a study even looking at genetics. So we have mice. These two mice actually are two genetically different mice, the mild and the severe. But what we found is that when we infected them with influenza, the mild have a lower diverse microbiome. It didn't change as much. And these guys is the more severe. So you can actually have genetics play a role in the overall outcome of the microbiome to infection. So you can have microbiomes that are originally more diverse that then get more diversified or less diversified. And what we were trying to do is associate maybe that the early diversification is really the driver to the outcome. And so we were able to, other studies of it were able to actually then take that and associate the microbiome with even psychological disorders. So things like anxiety and depression. What they're finding is that the gut actually becomes inflamed and leaky, and those toxins are being leaked out into your bloodstream, and then they're going elsewhere, causing immune responses in your brain. So these inflammations could be driven by your leaky gut, could then be causing disorders in your brain. So, and then understanding that this brain-gut axis is really as big as it is. So they did a study where they found that the gut access, so the nervous system of the gut is completely autonomous from the brain. So it can work autonomously without the brain. So basically you have two brains. And that whole feed the brain, I think is really, really viable now. And I think that that's why diet really plays a role and your idea of health, and it's really driven not necessarily by your diet, but your microbiome. So what I wanted to know is that now we have this and we can classify these things, so can we use them for actual clinical purposes? So I collaborated with this group in France, in Lyon, where what we were trying to do is we were trying to stratify clinical cases where we could say, okay, these are children with influenza. How can we take it? Because everyone said, well, you can't use this. So a person comes in with influenza, they're already sick. So what are you prognosing? What are you doing? And what we wanted to say is, no, I think we can still use the classifiers if we stratify our cases. So what we did is we took all children that had symptoms less than 48 hours when they came to the clinic, immediately got the nasopharyngeal swab, and then we used that. And what we said was, can we use that time point when they first come into the clinic at 48 hours, can we use that to then tell if they're going to go home with a mild influenza infection or if they're going to be in the ICU with severe? And the answer is yes, we were able to do it. And we were able to do it quite well, to the point to where we just submitted an EU patent on these microbial signatures for prognosing influenza infection in children. And we're publishing it as well. So, and we've also moved on to do adults, which has been done actually for the first time here in Tacoma, which I ran 60 samples through David Hirschberg's lab, and we just got the sequence back today. So we're hoping to be able to do a little last-minute submission to the patent as well to kind of beef that up on the adult side. But the reality is that we do have proof of concept. I think it can work in certain scenarios where we can actually use this information and actually predict infections or predict disease outcome. So really that goes to the subject of you can classify them, you can do that, but how do you manipulate them, right? How do we control them? How do we move them into a way where we can actually use this? Because right now our only major line of defense is antibiotics, and I am part of the anti-antibiotic movement because after seeing studies, so I've been involved in fecal matter transplant studies. I've been involved in lots of these different antibiotic studies where we're giving them to monkeys, and what the outcome of what happens to your microbiota after you give them something like vancomycin is you not only change the microbiome, but you change the whole environment. I mean, you're going from aerobic to a completely anaerobic environment. So that means the communities are changing and everything's changing. So to go after one bad guy, to me, it's archaic. I mean, it's to the point of chemotherapy where we need to be able to look outside that box and find a different way to intervene, especially when the therapies are already there. I mean, so this was a study basically just kind of looking at the overall effects of all these different antibiotics that we use and what they do to our microbiome, and almost all of them completely deplete a lot of the big players that we actually are associating with mutualistic or good bacteria. Things like lactobacillus. There's about 3 promising therapies that I think. One of them that I've done some work with, a couple other ones that I want to do some work with, is the FMT. So fecal matter transplants is basically one way to deal with this. And then bacteriophage therapy, it's something that's been around since the 30s and 40s. Russia and Georgia still do it. But for some reason, it's never been picked up in the United States. There are 2 companies now that are doing it. And then what I think is actually an interesting way to move forward, which is the microbial modulator. So, you know, these bacteria, they produce factors and enzymes and everything that are communicating with our own cells. So if we can figure out what those are, then we could actually use them to our advantage to be able to trick our immune systems to thinking that everything is okay when it may not be. So the fecal matter transplants have actually been one of the best examples of the effect of changing the microbiome to improve health. And that was with Clostridium difficile. It's a major problem that causes inflammatory bowel disease, which is a chronic disease. And what they found was that individuals that had high levels of Clostridium were more prone to inflammatory bowel disease. And by giving them a fecal matter transplant, you basically mitigated that by removing the Clostridium dominant bacterium microbiota and changing it with a diverse one. I think this kind of plays a role. This seems to work really well, but this probably explains why your probiotics don't work, is because you can give yourself a gazillion lactobacillus, but if your community does not want lactobacillus or has no need for what lactobacillus brings to the table, it's not going to accept it into the community, right? You're not going to just allow some stranger to come in and wreck your community if it's not doing anything and just hanging out and taking from it and not giving anything back. And that's kind of really how you have to think about how this works. And if you do have, so say you do have one, I think it works for some people because they have the right microbiome for it, right? And if you do, then you're probably going to accept it a lot easier, and it's going to have an effect whether somebody that has like 100% Clostridium difficile is not going to be affected by lactobacillus unless you do something invasive like these fecal matter transplants. Question?
So how would you measure and how would you know if you have high Clostridium? Is that what you called it? Clostridium?
How do you know?
Is there a way to measure that? Or?
Oh, absolutely. Yeah, I mean, you can measure it through PCR means, because obviously we know what the microbe is, so we can target it that way. A lot of use has been done to try to. So I did a study to try to go after that. So we wanted to know if you were going to set up an FMT study, what would be the best specimen to take, right? Because obviously a biopsy is very invasive. Then you have the actual poop. And then you could also do a swab, a rectal swab. So we were like, okay, rectal swab's in the middle, most invasive is the biopsy, and the poop would be the easiest to take. So we looked at all three. And what we found is that if you wanted to understand what was going on in the whole digestive tract, the fecal matter is great. But if you really want to understand what's going on in the mucosa, like what's going on in your colon, then the rectal swab and the biopsy were more similar. So now you have all these studies, and when I presented this at this microbiome conference, people were like, I have children and we've been collecting fecal matter for all these studies, and I'm like, I don't mean it's not informative, I just mean that if you really want to understand what's actually going on in your mucosa, the bacterium to the cells, then it's a little bit different story when you're looking at, so you need to look at the right specimen, really. And yeah. So I mean, this has really been able to work really well. And it's even been FDA approved. So they're actually moving forward. What I think is an interesting one, which hasn't been explored as much, is the whole idea of bringing bacteriophage therapy back, which, at the end of the day, so bacteriophages are viruses that infect bacteria and kill bacteria. Some of them kill, and if they do, they're called lytic viruses. So these are the ones we want to go after, the ones that actually kill the bacterium. And then some integrate into the bacterium and kind of hang out. Which we don't really care as much about. But those guys are the ones that make the bacterium resistant to the lytic guys. So there is a play there. But at the end of the day, we can use these things because phages are very specific to the bacteria that they infect. Unlike antibiotics, which goes after anything based on gram-positive, gram-negative, and these structural characteristics, these things actually go after just the single bacterium. So what's great about them is that they're pretty much the most abundant thing on Earth. So they're organic. So it's about as natural as you can get because you really can't do anything. They're very good. They've been shown in clinical uses in Georgia and Russia, and there's 2 companies now that are doing it to try. They've already been starting to apply it to the food industry because they've realized, hey, we can just do phages for salmonella and we can put it on the food to preserve it, which is a lot more brilliant way of handling it than giving your animal antibiotics for 3 years before you slaughter it. And then basically one of the first clinical trials has actually gone through, and it's been shown pretty good results of using phages to go after things like Clostridium. So as an alternative. But I think that this one is pretty much the one I think is going to take the cake because you don't have to necessarily control the whole or change the whole microbiome. Because what we found is there was a study done by one of my collaborators, Nikki Klatt, who was involved in the FMT studies and the monkey studies. And she just published a paper that was amazing. So they looked at South African women and the vaginal microbiome of South African women. And they were looking at, BV positive, so this is bacterial vaginosis. These are people that have bacterial infections in their vaginas. What they found is that there was 2 distinct populations of women. One population had pro-inflammatory, inflammatory microbiome. One of them did not. And that had the biggest role on susceptibility of HIV. And this is what we're talking about, but the majority of them that had this pro-inflammatory microbiome were not BV positive. So this is this is what I was talking about, this underlying disease baseline that we're not recording because we don't think it exists because we all think we're healthy. But we only go when we're really sick. But at the end of the day we could be chronically unhealthy for long periods of time. And I think that that's what we're trying to associate with some of these cancers and stuff, is this long-term poor microbiome that is now leading to downstream effects. So if we can use modulators, which are the enzymes and the communicators between the bacterium, then we don't even necessarily need it. So when you do get sick, maybe we can use the modulators to control the bacterium just for a certain period of time so that you can get over your virus infection or whatever, so you don't get a secondary bacterial infection, and even also controlling some of the inflammation. So I'm going to kind of end this by going through what I think is really, a real understanding of the microbiome, which isn't just about you. It's about everybody around you and who you interact with and how this actually is shaped. How, when I'm talking to you, are we actually exchanging our microbiomes? Are my susceptibility by being with you, am I more susceptible, less susceptible to diseases? I mean, they're already starting to associate microbiomes with even your attraction to individuals. So it's really about how do we understand what we're calling this microbiome cloud that's around us, and then how does the environment affect that cloud to either allow us to be healthy or make us sick. So this is kind of a new paradigm that's been coming out over the past few years, which is really kind of taking in the whole entire environment, pollution, everything. The vectors, the animals that you interact with, your food, and really kind of playing a role that this all affects in some way your microbiome, which then, if it affects your microbiome, it's affecting your immune system, and then it's affecting your health. So can we now take this bigger picture, and that's sequencing environmental samples to find out what's in the air, and it's in our water, and everything else, because we're going to find out we're not going to like what we find. I'm going to tell you that right now. So really, I think that the drivers of all this really is us, right? I think some of the major drivers and the shifts that we've seen in diseases and everything else has really been mostly caused by us. Globalization, climate change, the antibiotic resistance of bacterium. This is all us. This is all us. Urbanization. All of it is now leading to, I think, more susceptibility to diseases, but also drastic changes in our microbiome, which I think at more rapid rates than we're normally used to evolving. And I think that that's also leading to a lot of the things. I mean, I study viruses because I study emerging viruses, and I want to know the animal reservoirs and how these things actually transmit and go from an animal to a human. And what we are finding, for better or for worse, is that these outbreaks are happening at a lot more higher rate. Now we had an Ebola outbreak in 2014 that just ended in last year, and now we have chikungunya and Zika now that have moved in the entire Western Hemisphere and pretty much have now invaded. And I can name about 6 or 7 more viruses that are knocking on the door right now. My arrow is now already in Cuba, and it's going to be coming right up, and it'll be the next one. So these things are going to come up in greater force, and there's modelers that have been basically shown there's going to be twice as many spillover events of these type of zoonotic diseases in the next 50 years. So really, I started to think about that and say, okay, well, now we have chronic diseases everywhere. We have all this stuff going on. So how does this affect really disease itself? So there's a nice map of Africa that kind of shows this really crude idea of these chronic diseases that basically infiltrate different geographic areas. And I said, well, what happened if you overlay the Ebola outbreak onto that? And what you start to see is, you can start to see that there's actually pictures probably where certain diseases are going to be more prominent than others because the populations are already susceptible due to the chronic diseases they have, whether it's HIV, malaria, diarrheal diseases, and that these could actually be significant drivers. And maybe if we took Ebola and we didn't change the healthcare system at all and we brought it to the United States, maybe the mortality would be 30%, 20%, and not 80%. I think that these are really what we need to understand is are these players that we already have, these chronic diseases, are they actually, these endogenous retroviruses, are they actually playing a role as well in our disease and health? And then, one of these things that's kind of more dear to my heart, which is the agriculture industry and the abuse of antibiotics in the agriculture industry. I think one of the two major take-home messages over the past 2 years I think we've really taken from this is not the fact that we use antibiotics in our food and maybe that makes the animals unhealthy and we probably don't want that, but it's more about we're finding that the antibiotics themselves are the same antibiotics that we use. And when you have an animal that's full of antibiotics and you kill it and then you serve it to me, I am consuming those antibiotics, and they've actually shown that the antibiotics from meat can actually change your own microbiome. So you're giving yourself low doses of antibiotics by eating your antibiotic chickens and your cows and your pigs. The other one that was really big to me was that these antibiotic-resistant bacteria, things like MRSA, are basically being shown, in the clinics. Oh, this is all a clinic problem. This is because we sterilize in clinics. A paper came out that shows the primary source of these MRSAs being spread is coming from our agriculture production. So the handling of the meat itself. So what's happening is there's just a spread of these bacteria resistance going through our animal communities, which aren't causing disease, but they're actually then being handled and spread by us. So really, we're driving and we're spreading this whole thing through our own food sources. So yeah, we are what we eat. And then kind of the last one, which is what we're really trying to get at, I think some of the research is really trying to explore more, is this whole microbiome cloud, and this idea that we are actually like Pigpen in a cloud of microbes that are just being shed, and we walk around, and what we interact with, so whether you're in an urban setting or a rural setting, these are all players in your lifestyle, who you hang out with. These are all drivers in what's going to shape your microbiome. And if this is the case, then we can start looking at microbiomes at the population level and start to understand what the societal drivers or even are. And maybe that we'll find that there's some underlying societal drivers that are actually influenced by the microbiome. Yep, and that's pretty much all I have. Any questions?